Today we are going to have a lesson or our lecture on glycogenesis and to have an overview of this. But before we get into the overview, what are you really going to get out of this lecture? First, you're going to be able to know what glycogen is and be able to describe its structure. And then you'll be able to understand what glycogenesis is, what causes glycogenesis as we progress within the lecture. You'll be able to describe also the process by which the body forms glycogen and how the body uses and yes, those are basically some of the things, and finally you'll be able to understand some of the conditions or diseases or disorders that are related with glycogen storage in your body. I think we can go ahead and have the overview. So glycogenesis is basically, it is also called glycogen synthesis. That is another word. You can see these two words here, glycogen and then the synthesis. So we have glycogen, which is a molecule. To define or maybe to talk about briefly, glycogen is one of the molecules that is a primary source of energy in your body, especially during high intensity exercise and fasting. Glycogen is a storage form of carbohydrate in your body. Your carbohydrates in the body are stored in the form of glycogen, especially the excess one. So synthesis, this word biologically or scientifically means making out something. So these two words here can form glycogen synthesis, means the body making up glycogen. So let's look into some of these things. What brings in glycogen synthesis and under what condition? Under what condition do your body start synthesizing this molecule called glycogen? First, glycogen synthesis is stimulated by feeding, and then hyperglycemia and elevated insulin levels. So these three factors here are interrelated, that if you feed, you eat your food, possibly let's say bread, and you've eaten your bread or portion, any source of carbohydrate, your blood levels increases, your blood glucose levels increases. And when your blood glucose levels are high, that's what we call hyperglycemia. So that condition stimulates your body to secrete the hormone called insulin. Hormone insulin is secreted by the beta cells, the isolates of Langerhans in the pancreas, and its major role is to convert the glucose into, it aids in the conversion of glucose into glycogen. So that is basically what stimulates this process of glycogenesis. Going ahead, we look into this, that liver can store up to 100 grams of glycogen, which will last you, or which will take you 10 to 15 hours worth if you're resting. So let's say you're just resting, you're not playing football, you're not running, you're not jogging, you're not doing any other activity, but you have your glycogen stores. 100 grams can take you for 15 hours or 10 hours in just that range. Then the total muscles contains up to 500 grams of glycogen, though some can be found in the kidney and also adipocytes. So in this simply means we have the major sites where you can find glycogen: the liver, the muscles, kidney, and adipocytes. Then to getting into the structure of glycogen, a glycogen is just a linear stretch of glucose residues that are connected by alpha-1 glycosidic bonds with the branchings that are attached through alpha-1,6 glycosidic bonds. We'll get into more details about how this bonding of alpha-1 alpha-1,6 come about as we get deeper into the lecture. So it is just like an entire tree shape. It has a lot of branching. As we say it in this statement, that the branches are formed by the alpha-1,6. So on a linear chain, there are branches that forms up. Just imagine a tree having branches, and that's actually what we call, that's what can help you imagine what glycogen looks like. So each linear stretch, contains approximately 13 glucose residues, and it will carry about 2 branches of the same length that are attached in the space of about 3 to 4 residues, just like we can see in the slide. So going ahead into the structure, this is an illustration of what it is. When we look at these blue parts, the blue one is a linear stretch, and then there's also this dark one. From if you can see from the right side of the screen, there's what we call glycogen in here joining up, and there's also another linear stretch. Now the blue one, and where we call a branching, is where you can see where this blue one joins with the black one, and that is actually what you call the alpha 1,6 glycosidic bond. The alpha 1,6 comes in a way that on this black one, and the bonding is on carbon 6 of the glucose molecule, and on the blue one up, there is a bonding of on the first carbon of the glucose molecule. So I think hopefully that is understood, and we can go ahead to the next one. Now there are some steps in this process of glycogenesis in the synthesis. First is the activation of glucose, initiation of glycogen synthesis, and then elongation of the chain and glycogen branching. It's very important for us to have this glycogen branched, because this glycogen branching helps in increases the solubility and helps in the storage of these glycogen molecules. So makes it so easily stored by the body uh, in the tissues that stores them. And now right on the right side here is a summary of everything, the whole process. So you don't need to worry by looking into it. We will go step by step. And now to begin with activation, this is another illustration of how you came up with what you call the glycogen molecule. Right from the side up where you see glucose, and the whole cycle goes until you form what you call the glycogen molecule down there. So let's look into the first step, which is the activation of glucose. This involves an enzyme called phosphoglucomutase. And phosphoglucomutase will first transform this glucose 6 phosphate to glucose 1 phosphate, which is then converted to uridine diphosphate glucose. And before we get into that, let's understand all this, where it all starts from. I believe we have all known the process of glucose glycolysis. Yes, on the process of glycolysis, there's a step you reach at glucose formation of glucose 6 phosphate, and on that there's what we call formation of glucose 6 phosphate. So this enzyme called phosphoglucomutase helps in, it is very important in what we call transfer of phosphate groups. So you're seeing that the glucose has been on, the phosphate has been on the 6th carbon, and on this time around it's transferred to the first carbon. That's why you call glucose 1 phosphate, and that is done by what you call the phosphoglucomutase. And on formation of this glucose 1 phosphate, there's a there's a molecule called uridine phosphate triphosphate. There's a uridine triphosphate. So on this, that uridine triphosphate will combine with glucose 1 phosphate, and what it does is that it helps in getting one of the adding a glucose molecule to this glucose 1 phosphate, so that you can form the molecule that is called glucose, a molecule that is called UDP glucose. Sorry about that. So UDP here, to explain briefly, is this is the U is the uridine, DP is diphosphate, and in this reaction, when you react UTP with glucose 1 phosphate, the UTP releases itself with one of the group with one of its phosphate. That's why you have UDP. A phosphate had been on the glucose 1 phosphate. So with one of the phosphate also on the UDP, it will form, on the UTP will form UTP, and it releases a molecule called pyrophosphate. Pyrophosphate is basically two phosphate groups that are bonded together. So this reaction, however, is freely reversible, and the direction of its reaction is detected by glucose concentration. So the higher the glucose concentration, it will lead into the forward reaction that will form the UDP glucose. And then when the glucose concentration is low, it will lead into the reverse reaction, which is also going to be discussed later on, more of the breakdown of glucose molecules, the glycogen molecules, sorry. So let's get into the second step, which involves a molecule called glycogenin, as the synthesis requires a primer which is carried out by glycogenin. For you to have these reactions continue, you need a primer, meaning a molecule some some uh, preformed kind of glucose molecules. Uh, when you look at UDP on this side here, I hope you see my cursor. The UDP here joining up to glucose, to the to glycogenin, sorry. And glycogenin is formed, its structure has what we call an amino acid called tyrosine. Glycogenin is a protein. So the OH groups that projects from the amino acid tyrosine is bonded to the glucose molecules, and it releases uh, the UDP. So on this, we have the UDP glucose, which is bonding onto these glucose molecules to form this chain. And then after that, what happens is that the UDP is released and leaves on the glucose to join with the primer, and that joining occurs at the 1,4 linkages, meaning the glucose molecules that has been originally on the primer is bonded on the fourth carbon, and the one that comes is bonding on the first carbon. That gives us the alpha 1,4. That gives us the alpha 1,4 glycosidic bond or linkages. So this reaction continues until you form that primer that is ready for conversion. So for addition of more of this other glucose. So for this chain to elongate, to continue elongating itself up to the longest chain that biologically the system can contain, that is about 7 to 8 glucose units in just in its one chain. So that elongation is taken over by what you call the glycogen synthase. This synthase continues adding on more of these glucose molecules with the same bonding of 1,4 glycosidic bonds, and this is covalently bonded together. And the glycogenin, which is a part of the primer we had earlier, does not leave these glycogen molecules, but it remains attached to it. And so this reaction, as I said before, is catalyzed by glycogen synthase, which is also the key regulatory enzyme in this process of glycogenesis. This process too can be regulated, the glycogen synthase. So then the next step is the branching. After forming a long chain of 7 to 8 glucose units, the molecule has to be branched. The reason for branching, like I said earlier on, is important in helping us to the body to easily store this molecule, because the more the branching, the more soluble it becomes. So the more the solubility, the storage becomes so easy in your body. So something becomes also more compressed and compact than a very long linear chain of glucose molecules. So the branching here is done by branching enzymes. The branching basically means you form some branches. So branching is important because it increases solubility, like I mentioned before, and also increases the rate of synthesis and degradation of the large number of terminals. So this can make it so easily broken down, just like you have seen in this illustration. And on the right hand side, on the first image there, we are having the first branching around, then the UDP here continues. Once it has been branched, UDP, the glycogen addition, what you call, which is done by the glycogen synthase, continues adding on more of this on top of the branch formed, and also on the continuing chain where the branch has been formed on, until a point where it has to branch. That's like we said, the branches are formed around 3 to 4 units that have been formed. So then on the last one, you can see here on the right is now a branched unit, where you can see, I hope you can see my cursor, where we have the alpha 1,6 bond. This is the glycosidic bond that has been formed. All this, the original chain is what you're seeing looks blue, and then now the addition is this that looks white. It continues forming, and later on you see where this other one is branching. So after putting in a branch point, our enzyme called glycogen synthase comes and continues adding on more glucose units on top of the chain that has been formed, the branch chain that has been formed. So this is so continuous, and it occurs until the chain is really so long and stored in your body. And then, so this glycogen synthesis involves two types of reaction: elongation of the linear chain by formation of 1,4-glycosidic bond, which is catalyzed by glycogen synthase, and the formation of the branch points, catalyzed by the branching enzyme, like we talked about it earlier here. That on this side, and this reaction here, it's where glycogen synthase continues adding on the glucose unit. Each one of them is added using the 1,4-glycosidic bond. That is one of the major reaction. Then the second major reaction is this that we are seeing, the body forming the formation of the branch point, which is done by the branching enzymes. So I think that can be so much clearer now. So like we said, the three shorter chains that can be elongated by glycogen synthase. Each chain that has formed from at the branch point is elongated further by our glycogen synthase enzymes. So the summary, to have a summary of this glycogen synthesis, is that the synthesis of an activated precursor, UDP glucose, that is the molecule that is so activated and helps in this process to proceed. And then the second is initiation of the glycogen synthesis by glycogenin, the protein molecule I talked about that has tyrosine attached onto it, and that tyrosine, the OH group bonds with the first glucose molecules that forms now a primer. After forming on that first glycogen molecules, I think let me first take you back into this structure. Right here, what you're seeing is that we have the tyrosine. This is the glycogenin molecule, and this is an amino acid. Hopefully you're seeing my cursor. This here is the amino acid tyrosine, and on this point, the OH groups can react with the hydrogen group on the with this site of with the hydrogen group on the carbon 1 of the glucose molecules, and form this bonding and forms this very fast glucose molecules. This forms what you call the primer, and then after that we need to bring in, now what happened next here is the addition of glucose unit by what we call the glycogen synthase enzyme. So they add in there on this. When you look at this structure very well, we have this is the number 4 carbon. Starting from this side, this is number 1. Hopefully you're seeing my cursor again. Number 1 here, number 1 carbon, 2, 3, and then 4. So on this carbon 4, that's where the bonding forms in with this, which is carbon 1 on the next one, on the next carbon that is coming in. So this reaction continues in that way, and that is so basically what glycogen synthase enzyme does. Then to continue on, we have an elongation the chain elongation by glycogen synthase, and then the introduction of branches by the branching enzyme, like we said before, and then the steps repeats themselves. Uh, the exact steps it repeats themselves until the chain is long enough. And now, so after forming glycogen in your body, what else happens? That we get to see what happens. The process of degrading. Of course, we talked like earlier that this glycogen is utilized in the body. It's broken down in a highly intensive activities or during fasting and starvation periods. So these are basically the steps, and this is stimulated by exactly the reverse of the other reaction, starving during starvation period, and then hypoglycemia, and then we can the antagonist of insulin is a glucagon hormone, so they can stimulate this process of glycogenolysis, which is called the glycogen breakdown or degradation. So the steps basically are only three. First is the release of glucose 1-phosphate from glycogen. Second is remodeling of this glycogen substrate to permit further degradation, and then the conversion of glucose 1-phosphate to glucose 6-phosphate for further metabolism. So primarily, the primary reaction in glycogen degradation is cleavage of alpha 1,4 glycosidic bonds, and this is catalyzed by glycogen phosphorylase. This enzyme adds in phosphate, a phosphate group, and that is formed from pyridoxal phosphate, which is a PLP. We'll look at the role later on, but to mention it briefly, PLP, which is a pyridoxal phosphate, called, is a derivative of vitamin B6. So we'll get into that later. So on addition of this phosphate group to each of the glucose on the chain, it will result into the release of glucose 1-phosphate from the end of the chain. So the breakdown starts from the top part of the chain and it continues until it is broken down up to the branch chain. So looking into this as an illustration, before we go back to where the cursor is, so if the process of breakdown is taking place, this is where the phosphate, inorganic phosphate, is added by our enzyme, and then you release a glucose 1-phosphate molecule, which we, if we remember very well, was one of the intermediates during the formation, where we converted glucose 6-phosphate to glucose 1-phosphate in the process, in the presence of phosphoglucoamidase enzyme. So that's it for the beginning of the breakdown. So this reaction is repeated until the distance from the point is 4 glucose residues, at which time the branching enzymes begin to disassemble the branch points. So like earlier we discussed, there are branch points formed. So this reaction of breaking down of release of this glucose 1-phosphate continues until you reach 4 glucose units from the branching point, and then these enzymes won't be able to break down. This enzyme phosphorylase will not be able to continue with the breakdown of this glycogen molecule. So what happens is that the branching enzyme comes in and start cleaving the last 3 glucose units, which is a triglycerides, and transfers it onto the next one. Just like you're seeing in the on this arrow where my cursor is, that these 3 glucose molecules are cleaved off from this branch point and taken to the other branch point that has the 4 glucose units, and it results into an elongated chain on this side. If you can see the next one, and where we have the triglyceride transfer. So this triglycerides of 3 carbon is transferred on the next one and makes that chain at least much longer, so that our enzyme called phosphorylase can continue cleaving in and forming the glucose 1-phosphate molecules. And then at the branch point, this branch point can be broken by an enzyme called alpha 1,6 glycosidase. This is also a form of the branching enzyme that works at this point only, the exact point when we have left only 1 carbon or 1 glucose unit at the branch point. That's where this enzyme called alpha 1 6 glycosidase works. It breaks in the branch point, the exact branch point where the formation of the branch point took place earlier during its synthesis, and then you end up with a straight linear chain if this branch chain is broken down. So after breaking off this last branch chain here, we now have a straight chain that can be cleaved further by our phosphorylase enzyme until you have totally no glycogen in your body. So I think that's basically the process of breakdown. So to explain this, to have a question about this, this is a think and answer. You could explain why glucose 1 phosphate is the main product of glycogen degradation instead of a free glucose, and that has been discussed in somewhere. We'll get into it. So glycogen degradation involves removal of glucose, uh, which is glucose 1 phosphate from ends of the uh, glycogen chains and the disassembly of branch points. Since the phosphoglucomutase reaction is reversible, the glucose 1 phosphate derived from the glycogen uh, can be converted to glucose 6 phosphate. But we have a challenge that in the muscles we have glucose 6 phosphate, which is channeled, which the muscles lack an enzyme that can break this molecule. The enzyme is called glucose 6 phosphatase. So glucose 6 phosphate in the liver can be converted back to free glucose by this enzyme called glucose phosphatase. There's a spelling error that I'm seeing right there when glucose. So glucose 6 phosphatase converts this glucose 6 phosphate to free glucose. However, this cannot happen be in the muscles because the muscles lack this very enzyme. So that's exactly the answer to our question here. Sorry, it's not. I explain why glucose 1 6 phosphate is the main product of glycogen degradation or instead of free glucose. This, you can think about it. The answer comes from the degradation that the longest chain of our glycogen molecules is broken down by phosphorylase and form glucose 1 phosphate, the longest chain, and the only glucose molecules formed are those which are released from the branch point. So just like we saw on this, where you're seeing glucose 1 6 glucosidase, that's only one glucose molecule that can be released into your blood as glucose in the muscles. And so the rest here, when we check on this, that all this chain is going to be formed glucose 1 phosphate. That's why the major product of our glycogen degradation is going to end up being glucose 1 phosphate instead of a free glucose. So we can get further into the discussion ahead that now in muscle glycogen, in muscles, there is lack of glucose 6 phosphatase enzyme. And now this reaction you're seeing right here on this diagram is the one that only occurs in the liver and in the kidney, because the liver and the kidney has this enzyme glucose 6 phosphatase, which is capable of releasing a free glucose into our bloodstream. So when your blood glucose gets so low, it is only the liver and the kidney glycogen stores that is released into your bloodstream to help raise up your blood glucose levels. Otherwise, in the muscles, that glucose is channeled, glucose 1 phosphate is channeled and converted into glucose 6 phosphate, which is one of the intermediate in glycolysis. So on that process will continue that way and form pyruvate during the process of glycogen synthesis. Right here is an illustration of everything that you see here, and explain why glycogen, glucose glycogen, sorry, explain why muscle glycogen, this is supposed to be liver glycogen, contributes to blood sugars, while muscle glycogen, that does not contribute to blood sugars or glucose. So on this, uh, let's get into this understanding on the diagram. Right on my right hand here is the muscles. The glycogen is broken down to glucose 1 phosphate and then converted to glucose 6 phosphate, of which this glucose 6 phosphate is channeled into glycolysis, and then the Krebs cycle, and then forms ATP. And on the right side, we are having it in the liver, where it can be converted straight up to glucose and then taken back into the bloodstream. So this clearly explains why the muscles does not release glucose into bloodstream while the liver can. So the enzyme part of it is what explains everything here. So earlier we talked about PLP, which is the pyridoxal phosphate. We said it's a derivative of vitamin B6. So it serves as a prosthetic group for glycogen phosphorylase. So as glycogen phosphorylase adds in the phosphate group, this is part of it that helps in the process of phosphorylation of the glycogen molecules to form our glucose 1 phosphate. Then now, getting into this regulation of glycogen metabolism in fed and fasting state. The fed stage is like when you have eaten. The fasting stage is the opposite of the fed state, where you have nothing in the stomach and you have not eaten, possibly during the starvation and fasting, all that. So, like we said earlier, it is glycogen synthesis stimulated by feeding, hyperglycemia, and insulin, and this would be a repetition. So, glycogen synthesis is increased by glucose 6 phosphate. So, once you have glucose 6 phosphate and an abundant amount in your body, it is triggers or stimulates this glycogen synthesis. Meaning, once you have this glycogen synthesis enzyme in your body that is being activated, this whole process can now go ahead and form our glucose 1 phosphate, and then the whole process of glycogen synthesis continues. So, the regulation of glycogen metabolism in the fasting state, glycogen degradation by phosphorylase is stimulated by fasting. So when you're fasting, you don't have enough glucose levels in your body. There are certain things that are taking place in your body. One, you're hypoglycemic, meaning there is low glucose levels in your body, and glucagon obviously is activated. We have the hormones epinephrine, so this condition now increases phosphorylation of the cellular protein. So that process of addition of phosphorylation is addition of phosphate group. So uh, our phosphorylase enzyme will be able to continue adding on our phosphorylase, our phosphate, inorganic phosphate, onto the glycogen chain and form the glucose 1 phosphate. So you have to know that AMP, this is an adenosine monophosphate, is in a low energy state. It activates phosphorylase to its active form. So this is one of the things that activates our enzyme phosphorylase. Whereas the high energy glucose 6 phosphate, ATP, and in muscles creatinine inhibit phosphorylase. So that is one of the regulation. So a question can come that describe the regulation of glycogen metabolism during the fed and the fasting state, and this is basically what you have to describe about. So like I said earlier, we are going to discuss into these diseases of glycogen storage diseases, and they are basically characterized by accumulation of glycogen, and this accumulation results from deficiency in an enzyme that is important in these processes of glycogen synthesis or breakdown. So in some forms of glycogen storage disease, the structure of glycogen is formed abnormally. It's not a normal structure. So those that affect the liver are associated with hepatomegaly. Hepatomegaly is a condition where your liver is swollen, and then fasting, hypoglycemia, you're fasting and your body is hypoglycemic. Whereas those that affect the muscles are characterized by weakness, cramping, muscle atrophy. So this is basically it, and as we go through the diseases, we'll be able to discuss hepatomegaly, fasting, hypoglycemia, and how you can maybe get into nutritional management of them. So to start with what you call the type 1, the von Gex disease, this is the deficiency of glucose 6-phosphatase, meaning this enzyme here is missing in your body or is in very minute amounts that cannot perform its function as expected. So in this case, the glucose will fail to leave the liver and kidney, so it can't enter back the bloodstream. If we remember during our glycogen degradation step, after glucose 6 phosphate, we need glucose 6 phosphatase to break it down into free glucose and release into blood. So in that matter, meaning we are just going to remain with what? With our glucose 6 phosphate in the body. So that alone can result into accumulation and affect also accumulation of glycogen affecting its storage. And now the symptoms are subnormal serum glucose rise after glucagon or epinephrine. There's enlarged liver, which is the hepatomegaly. So once your liver stores a lot of this instead of it being released, it's a challenge because your body, your liver is already been packed with what is supposed to be released, and it results into that hepatomegaly, which is the enlargement or swelling of the liver. And then we have the kidney too can get enlarged, and then failure to thrive. So there's low energy levels that can be released for you, so you generally can't thrive. Then you can't exercise because this glycogen is what the body majorly uses for energy during intense exercise. So you can possibly not exercise because your body is not able to release into your bloodstream glucose so that you can be able to use when your ready glucose in the blood are low. And then we get into the reaction. You can feed with energy dense and frequent small glucose meals, the so-called nocturnal glucose, which can be helpful. This means during the night you can give in some foods, some energy dense foods that are having enough and can provide sufficient amount of glucose in the night, so that the whole process of the night, it doesn't allow your body to struggle converting uh, glycogen or using glycogen for you to survive, so that your bloodstream have sufficient amount of energy glucose that it can use as in your rest, the basal metabolic rates. This here is the illustration with the red there, where we're seeing 13. That's where that process is inhibited. And then we have the Cory's disease, and this one means there's no debranching enzyme. That is the alpha 1,6 glucosidase. So this results into accumulation of a branched polysaccharide. A lot of this accumulates in your body, and these symptoms are just similar to that of the Van Gog's disease, but they are milder. And one of the other symptoms is hypoglycemia. So once your body cannot be able to degrade this, you have some kind of hypoglycemia. Your body cannot have enough of the blood glucose at all times. Night feeding too is so good, and high protein diets can be very helpful because once your energy stores are low, the body has the mechanism of tapping into the secondary sources, which protein is an inclusive nutrient that the body uses for energy, just in case your glucose stores and glycogen are done. Uh, then there's Mercado's disease. This is the defect in the muscle glycogen phosphorylase, meaning you don't have the glycogen phosphorylase, which is in the muscle, and this is basically affecting the muscles. You can call it type 5. This is a disease. So glycogen phosphorylase is absent in this case, and you cannot break down the chain. So meaning the process of adding the phosphate group to the long chain so that you form the glucose 1 phosphate will not actually happen. So what happens next is that you have a huge buildup of the glycogen in your muscles. You can't utilize it. Whether you go for exercise, still you cannot break it because this enzyme is really not available. And then we get into symptoms. There's diminished exercise tolerance because your body cannot now, your muscles cannot release enough of this into your body. So you definitely can't tolerate enough of exercise. You have muscle crampings, due to the insufficient energy, and there's no rise in blood lactate after exercise. So that is basically the symptoms, and you can use this IV glucose to relieve muscle pain. So uh, I think that's it for the Mercados. That is basically happening in the muscles. There is what you call the Haas disease. It's more or less the same as the Mercados disease, but this specifically the Haas disease is for the liver, and the Mercados disease is for the muscles. So you can see the exact difference. It's still the same enzyme and the same bonds that have failed to break down the alpha 1,4 glycosidic bonds on the glycogen chain. So everything is still the same thing. It results into the huge buildup of the glycogen in your liver. There's hypoglycemia that your body cannot release glucose now into blood and acidosis as well. So symptoms are the muscle crampings on exercise, and there's no rise in the blood lactate. So the same treatment, you just give an IV glucose to relieve any muscle pain that you're having. So there's Anderson's disease, which is the type 6. You lack the alpha 1,6 glucan branching enzyme and can't make branches. So your body cannot make branches. And the symptoms are that you have an enlarged liver and spleen. Yeah, you have liver failure and cirrhosis. So there is no effective treatment that is known for this condition, and there is poor prognosis. Prognosis is the outcome of treatment, and is so deadly before the age of 5. I think a few of the children with these diseases actually survive. So I think that's basically all for the lecture. Thank you very much. Samuel.